A new study has settled a prolonged dispute within the medical community by showing that mutations associated with clonal hematopoiesis heighten the risk of atherosclerosis in affected individuals. In addition to the established risk factors for cardiovascular disease, such as high blood pressure, high cholesterol, diabetes, obesity, smoking, and lack of physical activity, another factor must now be considered: clonal hematopoiesis. This condition, caused by acquired mutations in blood stem cells, has already been linked to an increased risk of cardiovascular events. However, until now it was uncertain if clonal hematopoiesis was a cause or consequence of cardiovascular disease. Now, a new study published in Nature Medicine and carried out by researchers at the Centro National de Investigaciones Cardiovasculares (CNIC) resolves this critical debate by establishing clonal hematopoiesis as a new risk factor for atherosclerosis, the formation of lesions in the arterial wall that underlies most cardiovascular disorders. In a second study, published in the European Heart Journal, the CNIC scientists proposed the ancient medication colchicine as the central plank of personalized strategies to alleviate the effects of clonal hematopoiesis associated with acquired mutations in the TET2 gene. An adult person produces hundreds of thousands of blood cells every day. This high rate of cell division unavoidably entails the accumulation of DNA mutations in the dividing cells. These mutations are known as somatic mutations and are acquired, not inherited. Although most somatic mutations are innocuous, some give the affected cells a competitive advantage that allows them to expand and progressively accumulate, generating clonal populations of mutated blood cells, a phenomenon known as clonal hematopoiesis. These mutations had already been proposed as a possible risk factor for cardiovascular disease. However, the exact nature of the relationship remained unclear. Some earlier studies suggested that somatic mutations linked to clonal hematopoiesis contribute directly to cardiovascular disease and thereby accelerating the development of atherosclerosis. On the other hand, others proposed that it is atherosclerosis that causes clonal hematopoiesis by increasing the proliferation of blood stem cells and thereby generating a higher proportion of mutated blood cells. The Nature Medicine study clarifies the relationship between clonal hematopoiesis and atherosclerosis through a longitudinal analysis of data from the PESA-CNIC Santander study. PESA, Progression of Early Subclinical Atherosclerosis, is a prospective study of more than 4,000 apparently healthy middle-aged participants who have undergone periodic examinations using advanced imaging technology since 2010 to detect the presence and progression of asymptomatic atherosclerosis. The PESA study has already made very important contributions to our understanding of cardiovascular disease, and its longitudinal nature and unique characteristics provide an ideal framework for carrying out this important study on the relationship between clonal hematopoiesis and atherosclerosis. The researchers used high-sensitivity DNA sequencing technology to detect somatic mutations in blood samples and assess the presence and progression of atherosclerosis detected with non-invasive imaging techniques in the PESA participants. The study was a multidisciplinary effort involving specialists in basic science and cardiology, together with the specialized technical expertise of the bioinformatics, genomics, and clinical trials units at the CNIC. The results of the study clearly demonstrate that participants who had mutations linked to clonal hematopoiesis at the start of the study were more likely to develop atherosclerosis in the following years. On the other hand, the presence and extent of atherosclerosis had no influence on the expansion of mutated blood cells. These results indicate that the mutations contribute to the development of atherosclerosis, but are not a consequence of it. However, it remains possible that other factors, such as genetic profile or lifestyle, might modulate the effects of clonal hematopoiesis, and future studies are planned to examine this possibility. The results of the study have clear clinical implications and identify clonal hematopoiesis as a cardiovascular risk factor completely different from the traditional risk factors studied in recent decades. This novelty holds promise for the development of new strategies for the prevention of cardiovascular disorders. This study lays the groundwork for using colchicine for the personalized prevention of cardiovascular disease of carriers of mutations in TET2. But new clinical trials will be needed to conclusively demonstrate its effectiveness in these individuals. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.